Okay, good morning, guys. We won't wait anymore because we already lost like 7 minutes, so I won't wait anymore. And we're gonna start now. So for today, I might do more than 2 hours, and I might do I might reach 3 hours. Why? Because tomorrow I won't have so much time to be teaching. So the 2 hours of tomorrow they will be divided. I think 1 hour today and 1 hour tomorrow. And please for the assignments, please do them. And so it will be divided into you're gonna do your assignments and we're gonna do 2 hours every day, and Saturday Sunday we won't be doing a lesson. However, on Monday I really want all the assignments done, all of them during the week. And let's go. Okay. Okay. So yesterday we talked about speaking. We reached hemoglobin. So hemoglobin cooperative binding. Hemoglobin is an example of a protein complex with 4 subunits arranged in a specific structure. It carries oxygen in red blood cells. Fetal hemoglobin has a higher affinity for oxygen, while adult hemoglobin is more efficient at releasing oxygen. So one of the most important notes you should know for the IMAT is the third one, is that fetal hemoglobin has higher affinity to hold oxygen. However, adult hemoglobin, it releases more oxygen. Okay, fetal means the hemoglobin of the zygote or of the baby, of the newborn. So this is one of the things you should know for the IMAT. So high affinity of oxygen is fetal. Why? Because the fetus must have a very high amount of oxygen in his body because he's proliferating, his body is proliferating, is growing, so he needs much higher affinity of oxygen than an adult. Yeah. Hemoglobin cooperative binding refers to how hemoglobin binds to oxygen. When oxygen binds to one subunit, it changes the shape of the protein, making it easier for the other subunits to bind to oxygen. This cooperative binding allows hemoglobin to bind more oxygen efficiently, even at lower oxygen partial pressure. So the hemoglobin cooperative binding, why are we speaking about this term? This term speaks about how hemoglobin binds to oxygen. So when oxygen binds to one subunit, it changes the shape of the protein. So if oxygen binded to one subunit, it changes the shape of the protein. So when the shape of the protein is changed, it will be so easy for the other subunits to bind to oxygen. And by this cooperative binding of hemoglobin helps the cooperation of increasing the affinity of oxygen to bind more and more to the hemoglobin at a lower oxygen partial pressures. Okay, 1 second because someone is speaking on the group. Okay. They have been here. So, cooperative binding ensures a constant supply of oxygen to meet the changing needs of the body. This process is a result of hemoglobin's quaternary structure. The subunits of hemoglobin work together much like ingredients in a recipe to transport oxygen throughout the body. Hemoglobin cooperative binding is not a main question in the IMAT. However, you might just have to know that the node of the fetal hemoglobin, and you might have to know that the affinity hemoglobin has a higher affinity to hold or to catch carbon monoxide than oxygen. So one of the things you must know that hemoglobin has a higher affinity to catch or to hold carbon monoxide than oxygen, and that's why people say, or like scientists say, that smoking kills, because if hemoglobin has a higher affinity to be able to catch CO more than oxygen, this is a huge problem when you smoke. Okay? So we were speaking about the cooperative binding. We say that the more the cooperative binding happens, the higher the efficiency of this hemoglobin helping in catching more oxygen and making the blood more oxygenated. And hemoglobin is a protein, as we all know. So protein only functions in structure, in the main structure. So the main structure which hemoglobin functions is quaternary structure. So the function of hemoglobin, which is the cooperative binding, which is the function of hemoglobin, starts at the quaternary structure of hemoglobin. A globular and fibrous proteins, fibrous or you can say filamentous, the same, the one that I explained yesterday. So two main categories of protein structure. Globular proteins, spherical shape and soluble in water, perform various functions like hemoglobin or lactoglobulin, or immunoglobulins or antibodies, whatever. Fibrous proteins are long and slender shaped and soluble in water, provide structural support like collagen and keratin. We spoke about them, we explained them in details yesterday. Lipids. We have simple lipids and we have complex lipids. We have triglycerides, waxes, and the complex are phospholipids and steroids. Now, the triglycerides we have are made up of one glycerol molecule and the three fatty acids molecules. And the fatty acids, you should know that they can be saturated or unsaturated. One of the lipids that you have here is one of the main lipids that makes the cell membrane, which is the phospholipid. The steroids lipids are mainly hormones. The triglycerides, which is the glycerol and fatty acids, are kind of lipids that are digested in our stomach. And now we will see what does saturated fatty acid mean and unsaturated fatty acid mean. Lipids like fats, waxes, sterols, and certain type of oils. They are characterized by their ability to dissolve in non-polar solvents. Why? Because lipids are hydrophobic. So easily dissolving in non-polar. Lipids do not dissolve in water. As we said, the light dissolves light. Polar substances have an uneven distribution of charge and dissolve in polar solvents. Non-polar substances have an even distribution and dissolve in non-polar solvents. And this we know. We spoke about all of these. Lipids serve an important energy store. As we said, that it's not the main, or let's say the primary energy source in our body, but it's a very important energy store since it holds like 8 to 9 kilocal per mole. Fats and oils are composed of glycerol and fatty acids. The body can break down fats and oils to release large amounts of energy. The body can break down fats, oils, lipids. How can the body break down these fats? Using lipase, enzyme, and using bile from the liver. Lipids are also structural components of cell membranes, helping to maintain membrane integrity and fluidity. One of the main components of cell membrane is lipids. If you see the cell membrane, all of this membrane is made up of lipids, and then we have another layer of also lipids. So if you see, lipids is a very main element inside the cell membrane. It allows the cell membrane to be fluid and get structure. And then it gets structure. Sterols, such as cholesterol, are important components of cell membrane. Sterols, they sit inside the phospholipid bilayer here. Let's see here. Here the sterols position. Okay. Sterols act as precursors to hormones and other signaling molecules. Waxes are used for waterproofing and protection in plants and animals. So waxes are used for waterproofing and protection. Triglycerides, like fats and oils. Phospholipids are the main component of cell membrane. Carotenoids, which serve as pigments in plants and animals and provide protection from UV radiation. So lipids can help in the integrity and the fluidity of cell membranes. Lipids can help protection, waterproofing, protection from UV radiation, and all other and the energy source. So they have so much functions in our body and in any plant or animal cell. Now we're going to speak about the saturated fats. Saturated fatty acids have only single bonds between the carbon atoms. Fats or fatty acids are usually made up of, as we said, of a chain of carbon and hydrogen. And this long chain can be saturated and unsaturated. So of course, saturated, as we know in chemistry, means only single bonds, and unsaturated, you might find double bonds between this chain. They have the maximum number of hydrogen atoms and are solid at room temperature. Examples of saturated fatty acids like palmitic acid and stearic acid. You should memorize this note. Saturated fatty acids, palmitic acid, stearic acid. They are commonly found in animal fats. So this is for memorization. Saturated fatty acids include palmitic acid and stearic acid, PCS. Okay, and they are commonly animal fats. Unsaturated fats or unsaturated fatty acids have one or more carbon double bond. The chain of the unsaturated fatty acids might be like this, or it might be like this. Okay, so they will have this group, the double bond group. They have fewer hydrogen atoms and are liquid at room temperature, like oleic acid and linoleic acid. They are commonly found in vegetable oils. So the saturated are found in animals, the unsaturated in vegetables. Saturated are stearic and palmitic acid, unsaturated are oleic and linoleic acid, found in vegetables. Saturated and unsaturated fats. The degree of saturation affects the melting point of lipids. Saturated lipids have higher melting points than unsaturated lipids. Strong interactions between saturated fatty acid chains make it difficult for the molecules to move, resulting in a solid state at higher temperature. Double bonds in unsaturated fatty acids create kinks in the chains, preventing close packing and lowering the melting point. So saturated lipids have higher melting points than unsaturated. Why? Because unsaturated usually the chain is like this. However, the saturated, this is the chain. This is the chain. Okay, these are the double bonds. Here there are double bonds. So this double bonds, they might create kinks or might create branches. And this branches, it prevents close packing and it lowers the melting point. Okay, so higher melting points for saturated, they have high melting point, and the unsaturated, they have a low melting point. This for memorization. Why? Because they have kinks, so it have a low melting point and high melting point. I'm gonna explain it in an easier way. If we have here this chain and we have this chain, and we want to melt this kind of solid, okay, we want to melt it. So when you come to melt, you will find out that this is super easy to melt, and this is much harder because we have the king. Okay, because here we have the king, so it needs more time to melt. However, here you can just cut so easily. Okay, so that's the reason why we have a high melting point in the linear, and we have a low melting point in the king, in the double ones. And of course, there's another thing, that if we have more than one chain together of saturated fatty acids, okay, and we have more than one chain together of the unsaturated, the kinks. So which one of them do you think is much stronger bonds between them, much stronger interactions between them? Of course, this one. Why? Because there's no difficulties or there's no blocks between them to not make them strong. And this is also a main reason why the melting point also is higher here. However, here, if you see, there's a very huge distance between the interactions and very hard for them to be strong enough as the linear ones or the saturated ones without the double bonds will lead to a lower melting point. Okay, so the more the saturated, the more the strong. Okay, so high saturity, high strength. Of course, strength of the molecular bonds between them. Lipids can be classified as saturated, monounsaturated, or polyunsaturated. Monounsaturated or polyunsaturated. Saturated lipids contain only saturated fatty acids, means there is no double bonds. Unsaturated lipids contain one or more unsaturated fatty acids. And monounsaturated lipids have one double bond. Polyunsaturated have two or more double bonds. Okay? So because mono means one, poly means more than one. And when we say unsaturated, we are typically meaning double bonds. So it just depends on double bonds, on the functional group we have. The degree of saturation in lipids affects their function and their association with heart disease risk. More double bonds in lipids lead to increased fluidity of cell membrane. Saturated and unsaturated. If you see all of the carbons here, between them there's only single bond. However, the carbons in the unsaturated have a double bond on the third carbon. What does this mean? This means that this is a mono unsaturated lipid or fatty acid. Why is it mono? Because we just have one double bond. However, this is a monosaturated or saturated. We cannot say saturated because it cannot be too saturated. It's just saturated. Triglycerides are a type of lipid in the body. They consist, okay, so guys, triglycerides are a lipid in the body, but they are not the main lipid in the cell membrane. Okay, so triglyceride does not have nothing to do with the cell membrane. Cell membrane is created by phospholipid and by glycolipid. Triglycerides have nothing to do with the cell membrane. Okay, they consist of 3 fatty acid molecules bonded to a glycerol molecule. The shape looks like this. Here we have a glycerol. Here we have a 3 fatty acid chain. They might be saturated or unsaturated, these 3. The degree of saturation affects their physical properties. Excessive consumption of triglycerides can lead to obesity and various health problems. So we have a glycerol. How to memorize the shape of glycerol is just 3 carbon atoms, hydroxyl group, hydrogen, hydroxyl, hydrogen, hydroxyl, hydrogen, hydrogen, hydrogen. And then we have 3 fatty acids. So this one chain, second chain, third chain. So if you see that the fatty acid here ends with the carboxyl group, the cool group, this one, because it's a carboxylic acid, and it have R1 radical 1, radical 2, radical 3. It depends, it's variable. And then when we add them together, one H2O molecule is released. Okay, so this gives or this means that we are undergoing a dehydration or condensation reaction. So it's not just in glucose, it's not just in amino acids, peptide bonds, it's also in lipids. Ending up with this structure, and of course, because we are doing dehydration reaction, so it will lead up to the loss of 3 water molecules. Why 3? Because this bonds with this, this bonds with this, this bonds with this. So here we have an H2O, 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 ending up with. Okay. Saturated fatty acids, we have stearic acid, cis oleic acid, because they have a double bond, it's an unsaturated, and transoleic acid, they have a double bond. Now, both of them are oleic acids, but these 2 are isomers. Okay? So isomers mean that they have the same exact molecular formula, but they have different structural formula, okay? So here we have cis oleic acid, because we have the hydrogen and the hydrogen in the same direction next to each other. However, the trans oleic acid, one from here and one from here in opposite direction, makes it trans. Triglycerides are stored as the main form of fat in the body. They are also found in foods such as vegetable oils, butter, and lard. Triglycerides serve a crucial source of energy for cells. Glycerol can be converted to glucose for energy. Fatty acid molecules are broken down into ketone bodies for energy. So you should memorize this too so well. That the glycerol can be converted to glucose for energy. So to produce ATP, we use the triglyceride. The glycerol from the triglyceride can be decomposed and converted into glucose. And then the glucose will undergo glycolysis, reaching the ATP molecule. However, the fatty acids, when they break down, they form ketone bodies, and these ketone bodies undergo a kind of process to produce ATP. Okay, so each part of the triglycerides undergo a certain process in order to end up with a good energy source of the body. Okay, I'm gonna stop and make sure everyone. Okay, chat. Okay, perfect. All good, perfect. Okay. No way. Okay, we reached the phospholipids. Here is the most important. No, still here. Yeah, here. Triglycerides play a role in the structure and the function of cell membranes. Okay. Okay, so there was a question in the IMAT that said that triglycerides does not are not inside animal cell membrane, does not exist there. So let me just make sure, because I remember I solved it. Yeah. Okay, don't take this into consideration. Look, they can be they can play, yeah, it says play a role. Okay, take it into consideration then. Okay, triglycerides can have a small role in the structure and the function of cell membrane, but animal, because it was a question on the IMAT and it's so tricky for a person to think that triglyceride does not have to do nothing, or does not even exist in the cell membrane. So animal cell membrane, take it as a note, does not consist of triglycerides. They just have phospholipids, glycolipids, glycoproteins, hydrophilic carbohydrates, these things. So So guys, animal cell membrane does not consist of triglycerides, but they are a major component of the phospholipid bilayer. Fatty acid chains and triglycerides contribute to the membrane fluidity. Don't take this into consideration. Take it out. Focus on this. Okay? These first two, don't take them into consideration. I think triglycerides can play a role, but I think it does not have to do nothing with the animal cell membrane. Maybe another cell membrane, but the moment that they specify in the IMAT about animal, just say triglyceride have nothing to do with the cell membrane. I think if they spoke about other kind of cell membranes, maybe it might do, but like animal, just take it out. These two, don't take them into consideration. Fatty acid chains and triglycerides contribute to the membrane's fluidity and flexibility. This fluidity allows cells to perform their functions effectively. Triglycerides have formed the basic structure. I'm not knowing why. Okay, give me 1 minute. I want to make sure about it. Um, so I think triglycerides and phospholipids, triglycerides and phospholipids, because here the slide said that we have, because guys, I'm not doing the slides, Sam is doing the slides, Yusuf is doing the slides, and I'm the one explaining them. So that's why sometimes I get shocked. So triglycerides, guys, and phospholipids, here it's written that a major component of the phospholipid bilayer. No, triglycerides are something and phospholipids are other things. And I think they have super different structure between them, and they have maybe only one thing inside which is common, precursor molecule, which is glycerol 3-phosphate, nothing else. Glycerol 3-phosphate, which a question might be asked, what is the precursor which is in common between triglycerides and phospholipids? It's a glycerol 3-phosphate. Anything else about these? No. Okay, this is correct. Triglycerides are found in adipose tissue, which stores fat. Adipose tissue is concentrated in areas such as the abdomen, hips, and thighs. Triglycerides are also present in the bloodstream. Blood tests can measure triglyceride levels. High triglyceride levels may indicate health issues like metabolic disorders and cardiovascular disease. Phospholipids are essential for cell membrane structure and function. They consist of a glycerol backbone, two fatty acid chains, and a phosphate group. The negatively charged phosphate group imparts unique properties to phospholipids. Phospholipids are key building blocks of cell membranes and help regulate cell transport. This is the shape of the phospholipid molecule. So yesterday we spoke about when we have a molecule which have both the hydrophilic and the hydrophobic character, we call this molecule amphipathic molecule. So it does have a phosphate group, which is this, have a glycerol molecule, which is this, and two fatty acids. One of them is saturated, which is this one, and one of them is unsaturated, which is this one. If you see the double bonds here. So these two are hydrophobic, and we said before in the amino acids, in the protein structure, in the carbohydrates, even in the lipids. Whenever we have an aliphatic molecule, the hydrophobic part is always in the inside, and the hydrophilic part is always for the outside, because hydrophobic, they are protected from water by the hydrophilic head. The head consists of phosphate group, and phosphate is a very electronegative and a very negative molecule, super negative molecule. Why? Because this phosphate group contains four oxygen atoms. And these four oxygen atoms increase so much the electronegativity in this head. The glycerol molecule is this, and then here we have the three. Okay, I think guys, when they said phospholipids, triglycerides consist of phospholipids, I think they only meant by structure. In the slide, they didn't mean by function, or they didn't mean by like the exact thing. Because here, when the phosphate group enters the triglyceride that we have here, this is a normal triglyceride, one glycerol. And no, it have only two fatty acid chains. It's diglyceride. No. Don't take this into consideration. It has nothing to do with it. Maybe just because we have a glycerol and fatty acid, they said that. But no, each one of them is something. So as we said, guys, we have one saturated fatty acid and one unsaturated fatty acid. Okay. So the phospholipid bilayer, which makes the cell membrane, is like one. So more than one of these, all of them will be set together, arranged in two bilayers, one up and one down. And these two bilayers are asymmetrical. They are not symmetrical, they are asymmetrical. Okay. Okay. Phospholipids are involved in cell signaling, blood clotting, and inflammation. They are found in cell membranes, nerve tissue, and the brain. Dietary sources of phospholipids include egg yolks and soybeans. Deficiencies in phospholipids are linked to neurological disorders and liver disease. Adequate phospholipid intake is crucial for overall health. Fatty acid chains in phospholipids are hydrophobic. These chains vary in length and degree of saturation. The hydrophilic phosphate group and glycerol molecules are polar and attract water. Phosphate groups are often attached to water to polar and charged molecules. Phospholipids are amphipathic, meaning that they have hydrophilic and hydrophobic characters. Phospholipids spontaneously form bilayers. In these bilayers, hydrophobic tails face inward away from water, while hydrophilic heads face outward towards water. Phospholipid bilayers form the basis of cell membrane and organelles, and these bilayers act as barriers, regulate transport, and facilitate cell signaling. Selective permeability allows certain ions and molecules to pass through while blocking others. Phospholipid bilayers contain other lipids such as cholesterol and various proteins, because if you remember when I was drawing, whatever. So here's the cholesterol. It's inside. Yeah. So proteins in the bilayer can function as transport channels, enzymes, and receptors. Proteins are found here. Okay. And are found here in this way. Draw another color. Found in this way, let's say here, like this, like this. The inside proteins are called integral proteins, and the outside proteins are called peripheral proteins. Phospholipid bilayers are critical for cell survival and overall organism function. Lipid bilayers can form vesicles through self-assembly. Vesicles are created when lipids arrange themselves spontaneously in an aqueous solution. Phospholipid bilayers separate the inside of the cell from the outside environment. Proteins and structures on the bilayer surface perform various functions such as transport and signaling. Extracellular and intracellular. Here we have the hydrophobic tail, here we have the hydrophilic head, and this is the phospholipid bilayer. Okay. Sterols. Sterols are unique class of lipids with a fused ring structure. They play crucial structural and biological roles in cells. Roles mean like being hormones, like being any other function in the blood. Sterols have a structure consisting of 4 rings, 3 6-membered rings and 1 5-membered ring. This fused ring structure is important for regulating membrane fluidity and permeability. Cholesterol is the most well-known sterol found in animal cells. Okay, cholesterol, we have to focus on it. One of the most important sterols found in animal cells. So, cholesterol is an essential component of the human body. Its structure includes a fused ring system and a hydrocarbon tail. Cholesterol is also amphipathic, means it have hydrophilic and hydrophobic regions. So now we have phospholipid, we have cholesterol, both of them are amphipathic. It interacts with the hydrophobic tails of phospholipids in the membrane. Cholesterol has maintain membrane fluidity and rigidity. Cholesterol serves as a precursor for the synthesis of steroid hormones. It is involved in the production of hormones such as testosterone and estrogen. Cholesterol is also a precursor for bile acids, which are essential for fat digestion. The liver synthesizes cholesterol, and it can also be obtained from the diet. Ergosterol is a sterol found in fungi and plant cells. Stigmasterol is another sterol found in plants. These sterols have different biological functions. Ergosterol and stigmasterol share a similar chemical structure to cholesterol, made up of rings. Sterols are involved in the synthesis of hormones and other biomolecules. They help regulate cell membrane fluidity and structure. Sterols play a role in the absorption and the metabolism of fats. They contribute to the overall health of cells and tissue. Waxes, as we said, they are waterproofing and protection. So they are complex lipids with unique physical and chemical properties. Their intricate structure contributes to their distinct characteristics. Waxes are hydrophobic and water resistant due to their high molecular weight. Their water repelling properties make them crucial for waterproofing and protection. Let's speak about waxes. Let me give an example. Let's say Vaseline. Vaseline is a kind of wax. So when you put Vaseline on your skin, and then you put your hand under water, you will find out that water is not entering your skin. The water is creating bubbles on your skin. So why? Is because they are super water resistant and they have very high molecular weight and they are hydrophobic. Waxes are in nature super complex lipids which are super hydrophobic and super non-porous. So when this happens, they hate water. So when you put water on your Vaseline and then you wash your hand, you will find out that this Vaseline is not getting off your hand. It needs an oil, it needs a kind of soap or an acidic medium or an oil to be able to be taken off their hand. However, if you just put water or try to clean it with water, it won't be clean. You'll just create bubbles because it's water. Nucleic acids are crucial biomolecules involved in transmission and expression of genetic information. They determine an organism's physical traits, cellular functions, and behavior. Nucleic acids are essential for DNA repair and RNA synthesis and protein synthesis. Deoxyribonucleic acid contains genetic information and is primarily located in the cell nucleus. This is DNA. Ribonucleic acid synthesized from DNA and is essential for protein production. Ribonucleic acid, okay, and we have a deoxyribonucleic acid. The only difference between them is here we have a NOH functional group in its structure, and here we don't have the OH functional group. Here we have an oxygen in its structure. Here we don't have an oxygen, so that's why deoxy. Purines and pyrimidines. Purines and pyrimidines are nitrogenous bases found in nucleotides. So purines are adenine and guanine and have a larger structure with fused rings. Pyrimidines are cytosine and thymine and uracil. They have single ring structure or nitrogen ring. Let's call them nitrogen rings. The adenine and guanine have two nitrogen ring structures. Here we have a nitrogen, here we have nitrogen, and we have two of them. However, the pyrimidines only have one ring structure. Thymine is present in DNA and uracil is only in RNA. Nucleotides are the building blocks and nucleic acids of nucleic acids such as DNA and others. So here if you see nitrogen ring, nitrogen ring, nitrogen ring, nitrogen ring. Pyrimidines, one nitrogen, one nitrogen, one. So as we said, T and A, as we spoke yesterday, have two hydrogen bonds. And as we said, that hydrogen bonds occur between HO, occur between HF, and occur between SM. A high electronegative atom and the low electronegative atom. High, low, high, low. And these interactions are very weak interactions. The hydrogen bond, I mean. Cytosine and guanine, they create three hydrogen bonds. HOHN, HOHN. The size difference between purines and pyrimidines is super significant. Why? Because one of them has two nitrogen rings, the other have only one. Purines pair more easily with pyrimidines due to their larger size. Okay. Base pairing is crucial for the structural stability and proper functioning of DNA and RNA. Understanding the differences between purines and pyrimidines is crucial for comprehending the molecular basis of cells. Purines and pyrimidines are fundamental to the transmission of genetic information. They play a vital role in the processes underlying genetic inheritance. Purines and pyrimidines are essential components of DNA and RNA. DNA, deoxyribonucleic acid, is the genetic material in living organisms found in the nucleus made up of chromosomes. It's a double-stranded molecule with a sugar phosphate backbone. The four nitrogenous bases in DNA are adenine, cytosine, guanine, and thymine. Base pairing occurs through hydrogen bonds. A pairs with T and C pairs with G. DNA is wrapped around proteins called histones, forming chromatin. At the end of each chromosome, we have proteins out of the telomeres. Of course, we have the telomeres to be able to protect them while dividing the telomeres. Telomeres just act as a protection or extension for the chromosomes because they divide so much during mitosis, like so much division. So for them not to be cut or end, the telomeres extend them. However, at the end of the whole chromosome, we have a kind of protein which is called the histone. And this histone um, are one of the most important proteins in the cell cycle, the histones and the cyclins. Cyclins and histones are kind of proteins at the end of the chromosome. Chromatin can further, so what is the difference between chromatin, chromatid, and chromosome? Chromosome is this. Chromatid is one of these. Of course, here we have centromere. Okay. And here the point that allows these two chromatids to align together or to collide with each other is called kinetochore. Chromatids are one of them, and these chromatids can also be called a whole chromosome. There is no problem. And what else? We have the chromatin. Chromatin definition are the non-condensed chromosomes. They look like this. Okay. So before being condensed, they are chromatin. In prophase, in prophase 1, they start to be condensed. We'll start with metaphase already condensed to be able to align on the equatorial plate, looking at chromosomes. So here they are in prophase 1. In prophase 1, they condense. And their name become a chromosome, not a chromatin. DNA regulation involves the compacting and accessibility of chromatin. Heterochromatin, tightly packed DNA, while euchromatin is more open and accessible. So heterochromatin is not a chromosome, but still a chromatin, but super packed, almost looking like a chromosome. It is like this. They are tightly packed. However, the euchromatin is like, you know, euchromatin is actively transcribed, whereas heterochromatin is usually not transcribed. DNA encodes information for protein synthesis and other essential molecules. Transcription converts DNA into RNA using RNA polymerase. Okay? RNA carries genetic information from the nucleus to ribosomes. So how the sequence happens? DNA to RNA, protein, transcription, translation. Okay. This transcription happens in the nucleus. Translation happens in the cytoplasm, specifically in the ribosome and the rough endoplasmic reticulum. And then the proteins part and the packaging and the functioning, whatever in the Golgi apparatus. Okay, let me just make sure everyone is okay. A precursor to triglyceride, I think. What did you say about glycerol? 3-phosphate. Yeah, so glycerol 3-phosphate, I was saying that phospholipids and triglycerides, in their structure, there is only one thing which makes them in common. The only common thing between triglycerides and phospholipids is glycerol 3-phosphate. It's a kind of group which is inside the structure of both of them together. Why does gene have no protein? Gene is on the chromosome. They allocate. Let me just try to share with you something. The chromosome looks like this. Yeah. Here we have the gene sitting on in their positions or in their loci. So the genes here are on the chromosome. The histones and the cyclins are at the end of the chromosome, are proteins associating with the chromosome. So that's why the genes are here. Both of them include in the chromosome. Okay, is everyone okay? Everything is clear for everyone? Today we only have 5. I want to ask something. Rafi Rahman, are you a girl or a boy? Ah, okay. Perfect. Okay, let's continue then. Why is Zoom still here? Oh, I have to share screen. Okay. Okay, let's continue. Translation. Translation synthesizes proteins using the RNA template. Proper gene expression and molecule production are essential for cell function. So if you look in the figure here, you will see that the ribosome having the mRNA in the small subunit and having the bigger subunit, the tRNA or the transport RNA. So what happens in translation? The translation here, we have the two number 1. Tools of translation, ribosomes, tRNA. What happens in the process? So it's just the mRNA comes in here, the sequence, and then a tRNA sequence opposing the mRNA. Let's say like it depends on the nitrogenous bases inside the mRNA. So here we have the ribosome with a big sequence and the small sequence. And then it starts to sit inside the site. So here we have amino acid, amino acid. So what happens? This ribosome, all of the ribosome starts walking, starts moving around the rough endoplasmic reticulum. So when it moves 2 steps, 1 step, 1 step, it jumps 2 steps. When it reaches here, the ribosome, it adds an amino acid. And then what happens? These 2 amino acids will jump to the second site. Okay? And then they will add another amino acid. And then it will keep jumping, and here it jumps until they reach the end with a very huge amino acid. So then these 2 subunits, after we have a very huge sequence of amino acids together, these 2 together, they break down, the 2 subunits, they break down, they be far from each other. And this protein, because protein is made up of so much amino acids, so this protein will be outside free. So when it is outside free, they start walking and they go to the Golgi apparatus. When they reach the Golgi apparatus puts them in a vesicle, packages them, and takes them and makes them functional because they cannot leave the cell only if they are functional. So the Golgi packages them and then allow them to leave the cell, packaged, functional, everything. Of course, by exocytosis. And if you see here, this protein, which is made up of so much amino acids, of course, between them we have peptide bond. Okay, perfect. Nucleotide is made up of sugar backbone, phosphate, and nitrogenous base. All of this is called the nucleotide. There's three of them together, not just this, not just this, not just this. The sugar is 1, 2, 3, 4, 5, 5 carbon sugar. Yesterday we spoke about sugars and we spoke about glucose. Sugars are carbohydrates, means they are glucose and fructose and these sugars, you know. So yesterday we spoke about their structure and we spoke about that glucose is a hexose. We spoke about that fructose is a hexose, means it has 6 carbons. Which kind of sugar that you memorized yesterday used to have 5 carbons? Let's see if you remember. Ribose. Excellent. So ribose, so DNA, the nucleotide I mean, consists of ribose sugar, should he write ribose sugar, ribose, what's going on? Nitrogenous base and phosphate group. The difference between a nucleotide and a nucleoside is just that it's the exact same. We have a ribose sugar, we have nitrogenous base. The only difference is we don't have phosphate. Okay, pentose means ribose. Means pentose is 5 carbon or ribose. Nitrogen containing organic base. Okay, ester bonds, phosphodiester bond. Okay. So here we are speaking about the bonds between the nucleotides. Of course, each nucleotide we have phosphodiester bond, and between the bases we have hydrogen bonds. So if you remember, we were speaking. This is the DNA. Okay, this is the double helix. Here we have nucleotide, 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 nucleotide. These nucleotides create the nucleic acids, which are the RNA and DNA and tRNA. So nucleotide, 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 nucleotide. Between them is a phosphodiester bond. However, between the bases, hydrogen bond. Okay. RNA, ribonucleic acid, single-stranded molecule. Why is it single-stranded? Because, as we said before, the nucleus does not allow any double-stranded molecule to be out of it. The membrane of the nucleus, the pore membrane of the nucleus, just allows single-stranded molecules to go out or to enter. It consists of ribose sugar, phosphate backbone, and 4 nitrogenous bases: adenine, cytosine, guanine, and uracil. Uracil replaces thymine in the RNA, which is present in DNA. Okay, so I'll say something that we're speaking about RNA. Yeah. So guys, you remember, or I don't know if you studied or if you reached here, but we have 2 kinds of cycles. The first one is the lipase cycle and the lysogenic cycle. It's the kind of cycle obtained or attained by bacteria to a host cell to be able to attack the host cell. So the lysis happens that the virus enters and then they start to do mitosis and to replicate in a part of the host cell. And then they be able to kill the host cell in this way. They start to proliferate inside. However, the lysogenic cycle, the virus enters and then they go to the nucleus and then they enter the DNA of the virus, enters the DNA in the nucleus, and they change, they modify the DNA in the nucleus to be viral DNA. And when the DNA is viral, this cell will become infected. So why did I speak about this too here? Because viruses, okay, usually have single-stranded DNA and RNA, and of course, it depends if it's retrovirus or if it's like other type of virus, but usually viruses have single-stranded DNA. So why do lysogenic cycle happens? Because how do they do it? How does the lysogenic cycle occurs? Is the main reason that they have single-stranded DNA, and as we said, that the nucleus just accepts single-stranded molecules. That's why it's super easy for them to just let their DNA inside the DNA and modify the DNA and kill the cell. So, yeah, RNA plays a central role in transferring genetic information from DNA to ribosomes for instance. There are different types of RNA: mRNA, rRNA, tRNA, miRNA. Messenger RNA carries the genetic code from DNA to ribosomes. It's the supernormal RNA, which comes after transcription, of course. rRNA is the ribosomal RNA. Since we, is the ribosomal RNA and forms part of the ribosome and helps in protein synthesis. So, tRNA transfer RNA and helps the transfer inside the ribosomes, brings amino acids to the ribosome during protein synthesis. So how tRNA, it starts to get the amino acids, okay, inside the ribosome to be able to end up with so much amino acids creating the protein. miRNA or microRNA regulates gene expression by binding to mRNA. This depends on the transcription and regulation of gene expression. The main RNA type is the microRNA. And it kind of binds to the mRNA. RNA plays a central role in transferring genetic information from DNA to ribosomes for protein synthesis. There are different types of RNA. We just spoke about them now. The same. So let's speak about ribozymes. Ribozymes are RNA molecules with catalytic functions. Catalyst, what does catalyst mean? Catalyst means accelerate the reaction, kind of molecule that accelerates the reaction without interfering in the reaction or changing the product. So ribozymes play catalytic functions. Ribozymes are normal RNA molecules, but they don't interfere in nothing. They just try to accelerate the reactions. These are ribozymes. So here we have the comparison, and we're done. We spoke so much about them. Let me see if there's anything new to say. Yeah, everything you already know. So we're done with the first slide. I mean, with this PDF. And now I'll tell you what will we do. Let me just see if you have any questions. Everything is clear? Yeah, you should know them. Let me tell you about them. So, stop codons, start codons, and I wanted to also speak about something. Yeah, and number of steps. These three things. So, UAA, all stop codons start with a U. You listen. UAA, UAG, UGA. Start codons are AUG, which is methionine, and number of steps of transcription and translation. Steps of the process, I mean. Here 3 and here 3. So these also you should know. Why you should know stop codons, I will tell you now why when we just finish. When I just make sure everyone knew everything. So, anyone have another question? Do we need to know APE sites? Yeah, of course. Ribosome have three sites, A, P, E, and they when each ribosome jumps 2 steps forward, the amino acid starts jumping from the A to the P and then from the P to the A in this way, in a sequence. Okay, so I want to ask Saad, you are a new student here, and I want to ask you if you have any questions. So I think you should watch the recordings, the ones of before. And if you have any question of the recordings, I want you to text me on the IMAT Alpha WhatsApp, and I'm gonna respond to you on any question you have. Okay? Perfect. And they are not really so much, so try not to pile them up and make sure that you know everything. Sustainable advanced start codon. Yeah, mRNA have the start codon, which is AUG. So of course the RNA will bind. The RNA usually binds to mRNA, and mRNA have the start codon, so usually it will bind to it, or else the ribosome, the inside of the ribosome won't be stable. But like, does it have a start codon? I don't think. I think it have the complementary codon of the start codon. Okay, so okay, perfect. So now 5:21. Okay, guys. Okay, someone needs me out of the class. Take 10 minutes break and we're gonna continue. We might continue so much hours now. So take 10 minutes break, and after 10 minutes we continue. You can leave, you can stay if you want. I'm gonna keep it open. Okay. So, so let's go back to studies. So now I'll teach you guys about mutations, and then we will continue in biology until we reach a good amount, let's say 6. And then after this, so we're gonna start with the slide in chemistry. We might do the 1 hour of tomorrow. Okay? So okay. So mutation. Excuse me, guys. We have 3 types of mutations. We have substitution, we have deletion, we have insertion or addition. The kinds of substitution are missense, no, okay, silent mutation or silent substitution, and nonsense mutation. The deletion is always frame shift mutation, and the insertion is always a frame shift mutation. Missense mutation means it changes the amino acid which is made, because we're gonna have 3 codons. We're gonna have three, yeah, each 3 codons will no, we're gonna take 3 by 3, making ending up with 64 codons. Okay, and each 3 makes a different amino acid reaching the end. So when we substitute one of the bases by another base, it might end up by changing the whole amino acid. Let's say it was AUG, and then we ATG, and then we substitute the T by A. It will end up in AAG, and then the whole amino acid will be changed. And this means that it's a missense substitution. However, the silent mutation, it does not change the amino acid, but it changes only the sequence of the protein, of the bases. So the sequence of the bases is changed, however, the amino acid is still the same. So constant amino acid or same amino acid, no. Okay, but different sequence. Nonsense mutation is when we substitute one of the bases, and it make us reach a subcodon. Let's say it make us reach a codon which is UAG. So when we reach a subcodon, this is called a nonsense substitution mutation. When we are drawing let's say any sequence, any amino acid, and when we reach the stop codon, we stop. We don't continue because we don't care about the end. The DNA sequence now it's stopped. Okay. The deletion is when we delete one nitrogenous base, and then let's say we deleted here at all, we didn't substitute it. The A becomes next to the G, and the insertion we add between them, let's say T, which is both of them are frame shift. So these are the kinds of mutations. And yeah. So is it clear for everyone? Perfect. Okay, another thing I want to speak about is I want to give a kind of summary of the evolution. So now I'm giving random notes, okay? So much topics together, okay? So don't be confused. Okay. So take these definitions into consideration. Population. Okay. Same species and same environment. Communities. Different species. Same environment. Ecosystem. Living plus non-living altogether. Same environment. This way, I guess, it's easier to memorize. Biosphere, everything together. So also try to memorize them in this way. SS, population, different species, same environment, community. Living and non-living in the same environment, ecosystem, everything together is the biosphere. This is a kind of way to memorize them easily. Okay. Another thing. Okay. I want to go for chemistry. It's not like chemistry, but I just want to write a sequence, also try to memorize it. So we're gonna start with, on this axis, we're gonna start with the weak. We're gonna reach the strongest. Alkane, alkene, alkyne, alcohol, ether, ketone, aldehyde. This is a continuing for the arrow, okay? Let's do this. Aldehyde, amine, amide, ester, anhydride, carboxylic acid. Here's the strongest. Okay, so if we start from the weak to the strong, alkane, okay, have, it's like this. So it's so easy to break. Alkene, more stronger bond, and these two atoms are so close to each other. So when they are so close, it's super hard to break them. Alkyne is three, so super strong. And then alcohol, okay, we have an oxygen, so it's more strong than three of them. Ether, we have an oxygen between two radicals, which is also so strong. Ketone, we're gonna have an O and RR. So it's which is so strong. We have radical, we have oxygen. Aldehyde, we're gonna have an O and the C, okay, and then H, or an R and then H. Okay, but why we put the ketone before the aldehyde? Because the ketone have two radicals. However, aldehyde have one radical and another H. H is so weak. So that's why, because of the H, it's not as strong as the ketone. Amine is the NH. Nitrogen is one of the most strong elements we have. Amide is NH, but the carbon is bonded to an oxygen, so it is also strong. Okay. Wait, I guess there's something Okay, continue. The ester is a COO, like this. We have the two oxygen. The anhydride, it's like carbon dioxide, just like this. We have two double bonds, just so strong. And the carboxylic acid is this. We have two oxygen and one double bond and one single bond. So the strongest is this anhydride, ester, amide, amine, and then aldehyde, ketone, ether, alcohol, alkyne, alkene. Alkane is super weak. Okay. Why amide is more strong than amine? Because it have an oxygen. Okay. Why the ester is more strong than amide? Because it have two oxygens. Why the anhydride is more strong than the ester? Because the ester have one double bond and one single. However, the anhydride have two double bonds and two oxygens. Okay. Why the carboxylic acid is more strong than the anhydride? Because it have a hydroxyl group and a double bond and an oxygen. So try to memorize this. Okay. Yeah, it's NH2, exactly. It is NH2, but I just meant the elements like N and H, but I don't mean I just meant like we have nitrogen and hydrogen, but yeah, it's NH2 as a functional group. Okay, another chat. Why is amine stronger than aldehyde? Okay, so amine here and aldehyde here. Okay, so amine is NH2 and aldehyde is ROH because they have a nitrogen. Usually anything that contains a nitrogen is much more stronger than just having an oxygen. And aldehyde have an H. Okay. Okay, you might tell me that okay, why amine is stronger than aldehyde? Aldehyde have an oxygen, but there is someone that's strongest, which is the ester, because it also have oxygen, does not have nitrogen. So because the ester and all of them, the other ester, anhydride, and carboxylic acid, all of them have two oxygens. However, aldehyde and ketone and alcohol and ether only have one oxygen. So N is stronger. Let's write it. N is stronger than one oxygen, but two oxygens are stronger than N. Okay. You're welcome. Okay. Another thing I want to say, try to screenshot if you want. Perfect. Okay. So okay, I'm gonna speak about this. Okay. So I'll say something in chemistry because I remember that in the questions of stoichiometry, I guess, I'm not sure. Of course, you guys solved stoichiometry, but since we won't solve questions anymore together, maybe just math and physics, like together on the live on the meeting. Why math and physics and the others no? Because the others is if I came to each one of you, and I explained the question, biology and chemistry is much easier to understand than math and physics. Math and physics, you must know how to solve, you know? So like, of course, in my resting days, I'm gonna do an hour of math and physics, and we're gonna do it live. However, anything other we won't be solving only at the end of the course. At the end of the course, we're gonna put like 3 hours, 3 hours, 3 hours. We're gonna stop topic and we will start solving sessions together. So about now the questions about of IMAT and Alpha. These questions that you're gonna take are not from the past papers. These are questions created by us. However, the past papers, I'm leaving them till the end. We will stop topic at the end because we're gonna finish by the topic and we're gonna just focus on the past paper. And yeah, so someone's texting. Let me just make sure. What teaching? Can someone please tell? I just see now. We're just writing notes, like notes I've written from before. I'm just seeing my notes. Okay. Let's finish bio, and now we are starting a new topic. No, not a new topic. We're gonna start a new topic. What time is it? We still have like 15 minutes to start. So in this 15 minutes, I'm giving some notes and things. Okay. Like kinds of ways to memorize things, sequences, comparisons, and things. Okay. So I remember yesterday there was a question in the questions that we created that speaks about crystals in chemistry and stoichiometry. So crystallization or crystal. So I remember the question speaks about diamond, and now I want to just speak about the three crystals that we have, the three main crystals that we have, which are diamond, graphite, and ice. So I'm gonna speak in details about each one of them. So if anything was asked, you already know. So let's first speak about the diamond. So as a structure, it's made up of carbon, it's a cubic crystal. I think its structure looks like this. I don't remember, but looks like a cube, okay? So how this structure is, it's like here we have so much carbons, repetitive units of organized carbon element, only carbon, it's made up of only carbon, creating this diamond. And you should know that one carbon atom bonds with four other carbons, with four, okay, there's a mess here, with four carbons, and creating crystals. And diamond is super efficient. Why? Because they are packed perfectly together. Okay. So diamond is made up of only repetitive units of carbon, organized shape, in a cubic crystal. Each carbon is bonded to four carbons stacked together. This is the diamond. Okay, until now, anyone have questions before we continue to the graphite? And of course, don't forget that we're going to speak about the 3D shape of geometrical shape of each element. And the diamond 3D shape is tetrahedral. So 3D shape is tetrahedral. Anything might be asked, you should know about it. So tetrahedral, this number one. The hybridization of diamond, hybridization is sp3 since it's tetrahedral. And yeah, that's all you should know about diamond. So any question? Why it is tetrahedral? Because each carbon is bonded to four other carbons. If you remember methane, It's out of this, but like I'm gonna give another example. Methane, it's a carbon that have here 4 H's. So we have 4 bonds. The moment we have always 4, look at this carbon, 1, 2, 3, 4. When we have always 4 bonds, the central carbon here is tetrahedral. When we have 4 bonds, tetra means 4 usually. So that's why the hybridization is tetrahedral. So that's all you should know about diamonds. We can go to graphite. Okay, perfect. Graphite. So it's the same of diamond. The only difference between graphite and diamond is that each carbon. So each carbon atom is bonded to 3 other carbons, creating flat flat sheets, and not a cubic structure. It's called a layer structure. Okay, laying on each other's, sliding above each other's. Call it whatever you want. Just know that it's just a kind of sheets and not a cubic shape. So the structure of it, it's not a 3D structure since it's layers. It's sheets. We said it's sheets, so it looks like this. Carbon, 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 carbon. In this way, they are bonded. This is the graphite sheet. So each carbon is bonded to 3 other carbons, and they create the flat sheets. What sheet? Why I wrote sheet? Flat sheets. So yeah, so they stack like a deck of cards. In a way, like they are above each others, like a deck of cards. And their shape is not a cubic crystal. Their shape is a hexagon. What else you should know? Yeah, that's all you should know about them. Okay. And I guess their hybridization is linear or sp2. Why? Because if you remember that NH3 used to be like this. No. Let's say NH3 and H, H, H. It looks like a triangle. This is trigonal planar. Here the same, so we have 3 bonds, 1, 2, 3. And here we have also 3 bonds, so this is also a trigonal planar. So now I want, and both of them are just made up of carbons. However, diamond is 1 carbon bonded to 4 carbons. Graphite is 1 carbon bonded to 3 carbons. Now I want someone to tell me what is the main difference. Let's say we have carbon, 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 carbon. What about remaining, what are you speaking? What about remaining valency of carbon in graphite? So it's a carbon atom bonded to another carbon. You mean like it still have lone pairs? Okay. Let me make sure if it's trigonal, planar, or pyramidal. One moment. One second. Okay. Yeah, I said it's a trigonal planar. Ah, you mean, what are you asking? You're saying that you did the Lewis symbol. I'm understanding what you mean. So you said that you did the Lewis structure of carbon, and it can still form one more bond. Okay, let's do it together. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. Okay. You mean here we still have a lone pair? Okay, so here we have 1, 2. Here we have 1, 2. Take this off. Here we have 1, 2. Here we have 1. No, here we have 1. Yeah. Here we have 1, 2. Here we have 1. Here we have 1, 2. Here we have 1. Here we have 1, 2. Here we have 1. Yeah, so it ends up this and this. This one here, it's gonna bond with this. So this will go out to form a double bond. And here we will have a 1, and here we will have a 1. So this will go here by resonance, and then it will end up here, and it will end up in the stream. I don't know if you understood. If you want, I can repeat it on Scratch. Just tell me if you understood. I think the electron which is left can make a double bond with the other carbon. But like it's a trigonal planar and it's sp2. And you should not know about this. You just have to make sure about this. Okay. Perfect. So, so we spoke about diamond and graphite. Now, Sayed, I want to ask you personally, what is the difference between graphite and diamond? Both of them are carbons. Like, let me tell you, like, let's say we have, okay, look here. All of you look here. Let's say we have carbon, so much carbon atoms, okay? And we don't know if they will happen, if they will become graphite or diamond. What happens? What are the characteristics that must be in the surrounding for the carbon to be graphite, and what is the characteristics that must be in the environment for the carbon to become a diamond? What do you think, guys? Does anyone have an answer? Bravo, excellent. So it depends on the temperature and the pressure in the surrounding. When we have a high temperature, okay, and we have a very high pressure, so diamond creates out of these carbons. However, if we had these carbons under low pressure, no, why am I putting a degree Celsius for pressure? So under low pressure, this means, and high and the low temperature, this means that we have the carbons will form a graphite. Why? Because usually, if you know that, like we as any geologist, find diamonds underground means find diamonds deep in the soil or deep in the earth, okay? However, graphite is found in the stems of the trees and like these things which are out for the surrounding. So the stems for the trees are not in a very high temperature like the diamond. Diamond is it's in a very high pressure, super high pressure because it's under soil and a very high temperature under soil. However, graphite, it's in the trees, it's in the normal surrounding that we're living in, and it's a very low pressure, a very good pressure. So that's why graphite is much more common and much more dominant on Earth than diamond. Because diamond, they are really hard to be formed. However, graphite can be formed anywhere. And that's why you see so much pencil. So is it clear? Okay. Now we speak about ice. Understood. Okay. I just want to add something that in the graphite, I said that we have layers of carbon. So just put in mind that these layers, all of them are super parallel. And of course, it's repetitive units of carbon atoms. Okay, now we're gonna speak about ice. So, we're gonna speak about ice. It's super easy. It's repetitive unit. So usually graphite and diamond repetitive unit of carbon. Awesome. We are speaking about crystals. Crystals are called repetitive units of something. An element have a repetitive unit. This is the definition of crystals. Organized repetitive units. So usually any crystal is made up of a unit of an atom which is being repeated. Okay? And I want to also add just yeah, I want to give just a small side note before we continue about the system. Let me write it. So okay. Yeah. I know it's in Italian. Let me just try to translate the word. Okay. Okay, I'm not giving the same. Let me try to make sure what is the name. One second. Yeah, they are called aperiodic crystals or quasi-crystals. Okay. So we have kind of crystals. Before we go to ice, we have something called quasi-crystals. Or you can say aperiodic crystals. I have the same definition of crystals are ordered or organized atoms forming a crystal. The only difference is that they are not repeated. If you saw that we have so much in diamond, so much carbon atom repeated. Here, no, they don't have repeated atoms, and they are not periodic. So this also take it as a note, quasi-crystalline. Okay. If you want an example of these, we have He crystals, Ng crystals, Zn crystals. These are naturally quasi-crystal. Naturally quasi-crystal. So now let's speak about ice. Is it clear? Okay. So we're gonna speak about ice. So since we said that crystal definition is a repetitive unit of something, which unit in ice you think it's repetitive as a crystal? Excellent. That's true. So ice have repetitive water molecules. So that's all you should know about it. So it's like H2O, hydrogen, oxygen, hydrogen, then oxygen, hydrogen. It's the same, like it's water, at the end of the day, this is water, yani. And it just becomes solid by freezing. So, and makes a crystal. So it's repetitive. You should just know about it that it's repetitive units. The crystal, which is ice, is a repetitive unit of H2O. Okay. So this about crystals. Really done. Now we're gonna speak biology about yeah. Okay. So now we're gonna speak in biology about the endosystem. So the endosystem, does anyone know anything about it? Can anyone, since you already guys got the base and finished the topic, like studying by yourself, so can anyone list for me what is the endosystem? Because I really want to know that you guys went through all these things and got 100%. Excellent. Okay, guys, I really want an interaction from you, not just from Sayed, because I'm here explaining for everyone, and you are already so much participants now. So everyone has to try to interact with me. So the nervous system, it's the system that involves endoplasmic reticulum, both of them rough and smooth. Uh, okay, it involves the nuclear envelope. It involves the Golgi body. It involves all the vesicles. It involves the plasma membrane. It involves the lysosomes. Lysosomes, if we remember, these are the kind of vesicles inside our organelles that kill and digest all the pathogens that might enter or the unneeded things in our cells. And their pH is 5 and they have 30 hydrolytic enzymes. So this is the endosystem. Just memorize is what is the endosystem made up of. Messenger system. Okay. So now we're gonna speak about the cytoskeleton. So the first kind of cytoskeleton, number one, what is the cytoskeleton? Cytoskeleton is everything inside the cell which maintains the shape and maintains the function, maintains the movement, and the support. What are the kinds or the types of the cytoskeleton inside our cells? Are microtubule shape, are intermediate filament, and are microfilaments. Microtubule shaped like the spindle fiber, and the spindle fiber is one of the most important cytoskeletons or microtubules that are in our cells, which help occur this cell cycle and help align the chromosomes. Their shape looks like this. And they have a positive and a negative gram. The positive is always from up and the negative is always from down. And what else? We can here always add 1 and we can here always subtract 2. 2, 2, 2, here we can add 1, 1 more. And this way the microtubule is done. The intermediate filament is like keratin in the hair. So it's all the filaments which help in the structure because keratin have no function, just help in the structure or in the support of our hair. Microtubule help in the function. So here support for the intermediate, the function is the microtubule, the shape in the intermediate, and the microfilaments look like have helical structure like this. They have, or like we can do like this if you want. And these microfilaments help are also functional, not just structural. Intermediate are more structural than functional. This microfilaments, they help in contraction of muscles. Not all of them, but only one of them, which is the actin. So if they ask, what is the actin? Actin is a kind of thin molecule and the basic unit of the muscle cell. So they help in contraction of muscles and they are helical structure and they are microfilaments. So the kind of actin is a microfilament. Clear? And what else I want to ask? Okay, I want to ask you also a question. Guys, do you think DNA is a micromolecule or a macromolecule? So DNA is a micro or a macro? Excellent. So just put a note here that DNA is a macromolecule. Yeah. Okay, let's see. Macro, excellent. So in microtubules, what is plus one? Yeah, these are the grams. These are the grams. You can here by the microtube, by these grams, you are able to add more filaments. You are able to take from this filament. It kind of say that it can be variable. You know. I just explained. So it kind of says that you can add more grams. You can add more molecules. You can take from it. It's like the microtubules can be variables. So now, okay, guys, do you know about evolution? Anything like evolution? How, like, what is evolution? The natural selection or these things, or you want me to explain them now? Do you have like problems with them? I'm gonna like we will have a whole PDF about it. We're gonna have a whole class about it. But do you think like it's better to understand it and explain it now? What is your base, guys, of evolution? Oh, it troubles us all. Okay. What do you mean started by the two types of wall in bacteria? What do you mean by this? Oh, you mean the positive gram? Yeah, yeah. Okay. So, okay, let's speak about evolution then. Okay. Okay, I wanted. Let me remember. Oh, perfect. Perfect. So I'm gonna teach about evolution now. It might take the time, but there's no problem. Okay, so focus, guys. It's the kind of topic which no one knows how to study it, to be honest, because it's so like somehow complex, but it can be studied. Let's go for it. So evolution, it's it's a kind of speaking and saying how did we update, how did we improve throughout the decade. So it's made up of types, and the selections are three. So we have the natural selection, we have the genetic drift, and we have the gene flow. So the natural selection, it's the highest frequency of certain alleles. Alleles have high fitness in a specific environment. The increase of frequency of an allele in an environment makes a huge success and allows the high fitness in the specific environment. So high frequency in certain alleles have high fitness in a specific environment. Genetic drift is allele frequency change randomly. So allele frequencies changes randomly. So it's kind of genetic drift. It's kind of surprise for the alleles. It was like this in this let's say this mom got this kid. So this mom have blue eyes. The kid out of nowhere he have um even if the dad have both of them have its dominant, which is the blue eyes. The kid out of nowhere he had brown eyes. So this genetic drift, it comes from nowhere in a random mode and changes the whole allele frequency. Gene flow, it's a new individual, okay, introduce new allele to a population. Let's say we have a generation, and then all the generation were, let's say, having Asian eyes. However, one out of the West came and married one of the generation, and then a huge different generation happened. So one individual introduced a new allele to a population. Now I'm speaking about a human, but it's most likely the evolution we speak about animals. So we're gonna like the IMAT only speaks or asks about animals and evolution, not humans. But I was trying to make it reach you easily. Okay, so these are the types. Now we're going to speak about the modes. Second point of natural selections, alleles have high fitness in a specific environment. Means this allele is very high percentage that all the generations gonna have. So the evolution modes are stabilizing selection, directional, disruptive, and balancing. Okay. So okay. So we're gonna speak about stabilizing selection. Okay, look, first I'm gonna draw the graphs and then I'm gonna explain, okay? Okay, one moment guys, because I'm trying to get the figures. Okay, perfect. Okay, so here we have like this. Here we have like this, and here we have perfect. These are the figures. Now, according to these figures, we're gonna see what does each mode mean. So, stabilizing selection, this type of selection acts to maintain the current phenotype in a population. What does this mean? What does this figure mean? This figure means that we have a specific phenotype in a population, okay? So what happens in stabilizing selection? This selection mode always try to make this exact phenotype, this one, always be increasing during all the generations to end up having this phenotype all over the generations. It favors individuals with intermediate phenotypes and selects against individuals with extreme phenotypes. So let's say individuals that have intermediate phenotype means, let's say the phenotype was AB, and let's say another individual which have only A, just super extreme. So it favors the one which have stabilizing. The rule you should memorize is it favors individuals with favors intermediate phenotype and against extreme phenotype. So this is the rule for stabilizing. Let me give you an example to make it more clear. Let's write the example here. Let's say the humans, the birth weight or the body size in a population. So it's like it includes the selection for optimal birth weight. What does like optimal birth weight means? Means it favors the intermediate birth weight and it's against the extreme weight of a human, the size of a human. So the stabilizing, from its name, it's kind of stable, a stable selection. This means the human cannot be let's say, like the population of humanity cannot be in the size of an elephant, you know? So this kind of examples speak about stabilizing selection. The examples that are really known in a species. Okay, like this species, this is its size. Or like let's say the species of ants. خلاص, their size is so small. So the selection of size of an ant is called stabilizing selection. Make me sure that you understood, guys. Okay. Here. Okay, directional selection. If you see, you can see two phenotypes. Okay. So this selection, write as a note that this selection favors phenotype at one extreme. So stabilizing is against extreme because it's stable. Everything which is stable in a population is stabilizing. Directional, it favors phenotype at one extreme. Okay? And it can, by the way, this can lead to a diversity in population. Why? Because it can change. It can give another phenotype. It can favor an extreme of these two. So it allows diversity. So when they might ask you which selection might allow the diversity, you're gonna say directional because it allows more than one phenotype. However, the stabilizing, it's only one and no diversity in it because the size is the size, the weight is the weight. Everything which is stable in a population, let's say the population of birds. All birds have beaks. So having beaks, this selection of beaks for birds is called stabilizing selection because all of them have the beak. You understood in what way it is stabilizing? Okay. However, let's say that let me give the same example in directional and try to show you the difference. So stabilizing, all birds, all of the species have beaks. This is stabilizing. When does this exact example becomes directional? Okay, some birds have big beak. Other birds have a smaller. Let's say eagles. Eagles are a type of birds, but they have bigger beaks. And we have some other birds which have smaller beaks. Why is this? It's because there is a directional selection happening here. Directional means we have one big, one small. So here we have two. We have here the big and here the small. So this shift in beaks and the change of beaks, as Darwin says, here we involve Darwin. So as Darwin says, that why do some birds have big and why do others have small? He said, okay, all of them have beaks. Some of them are created with big, some of them are created with small. This depends on their surrounding, on their environment. Some birds are not able to eat food, which is, let's say, like it depends on the food chain. It depends on the food, how like the food availability. Some of them need to eat from the trees. Let's say they must have a long one. Others, they are able to eat from next to them, close to them. So it depends on the food availability. So Darwin spoke about food availability and explained this directional selection according to the food availability. That's why we have more than one phenotype, and we favor the one extreme phenotype in them, and we allow the diversity, big and small. However, here we just have in this table only one. All of them have beaks. So you can say in a mode that stabilizing selection is more general, beaks in total. However, the directional is small and big, so it's more specific. Okay, until now, everything is clear, or you have any doubts? Clear, perfect. Okay, perfect. Disruptive selection. Disruptive selection is a type of selection favors individuals with phenotypes at both extremes. Okay, so if you see here, it's favoring only one extreme out of two extremes. Okay? It's favoring this one. Here it's not favoring, it's favoring an intermediate phenotype. Means خلاص, it's something which is stable, constant. Disruptive, it favors two extremes, here and here, and that's kind of way to understand the figures, you know? So the figures try to help you to understand. So disruptive is between the two extremes, this one here. It's favoring this part, this small part. Here it's favoring this colored part, here favoring this small part between the two extremes, and here is favoring all of this constant part. So we can write here as a note. Okay, guys, because it's really small, I'm thinking of deleting and starting from disruptive on a new one. So let's do this. Okay, perfect. So the disruptive is like this, and there's the disruptive. So the disruptive selection favors phenotypes at both extremes of the distribution rather than the intermediate phenotype. And it can so now we have two which speaks about diversity. The directional and the disruptive, both of them allows the diversity. However, the disruptive can create a new whole species, you know? Let me give an example. Okay. So let's stay speaking about beaks. Okay. Let's speak about beak morphology. So let's say eagles have this the big beak, and the normal bird have a small beak. So when this eagle have a big beak and the bird have small beak, as we spoke about the directional one, we said it depends on the food availability. Okay. So the eagles, which have big beak, have a feeding different. Both of them have different feeding niches. Each one of them has a kind of prey they want to eat. Each one of them has a kind of a kind of food that they would like to eat depending on their beak. So how is this happening? It's just because the disruptive selection, it's like one phenotype depends on the other. Because of this character of the structure of the beak, okay, the inhabitants to be eating a kind of food will be this. So when one affects the other, this is affecting the other, he disruptive selection happens. So like, did everyone understood? Or like, you think it's not really clear? Perfect. Humera, you're a girl or a boy? I'm sorry, but I don't know like the names outside if they are girls or boys. I know Saad is a boy. Humera. Nice name. Girl. Okay. Okay. Perfect. So as we said, let me try to give another example. Like small fish, they try if you search for small fish, you usually find them in fresh water. Okay. However, there's another kind of fish which have like bigger gills and bigger body and these things. You might find them in another kind of water. So it depends why those small ones are like can be in fresh water and the others cannot live in fresh water, because the small ones, because they have small gills, they are able to live in fresh, they are only able to live in fresh water. However, the others, they have big gills, they have more a bigger filtering system, so they can easily live in a normal water. So you know, like when you get a fish to the home and then you have to always change the water because they are small fish. So depending on the environment the um, is affecting the phenotype of small and big. You understood how? Like one of the characteristics affect the other. Okay? Okay. So you saw in what way I'm meaning disruptive? There's something affect the other, allowing a character to dominate in a species. Okay, now we're gonna speak about the balancing. Oh. So the balancing selection. balancing selection. We're gonna have. I remember we drew the. Let me first draw the graph. That's a kind of selection that maintains genetic variation. So it maintains genetic variation within a population by favoring the persistence of multiple alleles at a given locus. So balancing selection is kind of selection that maintains genetic variation within a population by favoring the persistence of multiple alleles at a given locus. Let's speak about about butterflies. So guys, butterflies are usually colored. You might see one yellow butterfly, the other is red, the other. So these butterflies, by their normally butterflies are colored. Why are they colored? According to the balancing selection. This kind of balancing selections, balance that this population, this species itself gonna be colored. You know? So it tried to persist or to maintain multiple alleles at a given place, at a given locus in a species. So the more the colors of butterflies increase, the higher the balancing selection is working in. Okay? So balancing selection try to maintain the genetic variation within a population. Okay? So you might not see the same color of butterfly always. In a species, you might find 10,000 color, and this is a huge example of balancing selection. Change of color in a very same species. They might have the same structure, the same form, the same eyes, the same organs, but super different color. So this is the balancing selection. So is it clear? Because we're done with the selections, and we're gonna go for the chemistry slide now. Okay. You give me 1 second, guys. Perfect. Okay. So let's go here. See the chat. Clear. Okay. Okay, guys, I'm gonna tell you something that we're gonna start now the chemistry of tomorrow. We might do an hour, we might do 2 hours, it depends. We're gonna try to finish the slide now of tomorrow. So tomorrow we won't have a meeting. You're gonna have Friday, Saturday, Sunday without studying, without live classes. But we're gonna open a new Zoom meeting, which is the chemistry one. Of course, we won't try to